right, it looks like things are working. We'll just give it a couple minutes here. I started a little early. I do have to say, though, that you guys are going to be keeping me from something important. So, I mentioned in my little video last night, I got this new toy. I am eager to head over to the lab and start playing with it. I mean, look at this thing. It's like a microscope objective built into a camera. So, yeah, I got to get over there and get up close and personal with some spiders and take some pictures. If spiders horrify you, you might not look at the blog for a while. Nah, I probably won't put them on the blog because I get so much pushback if I do that. But anyway, lovely new toy. I'm eager to try it out. Okay, set that over there. All right, what do we got for viewers? Okay, we're up to a whole 4 people. Oh well. Yes, it's a Canon T7i. It's just a nice little, commercial home camera sort of thing. And I think I've spent more money on the objectives for it than I have on the camera itself. Yeah, that's definitely true. So, all right, so let's start talking. So did everyone do the homework and read the paper? No, you don't have to. There's no homework. So, is it heavy? Is it? I don't know. Is it? Not particularly. It's not like a point and shoot, but it's fairly easy to get around. At least my old frail muscles can still hold it fairly steady, so it must not be too heavy. Okay, as I threatened, we're gonna talk some more about this really cool paper here. So again, if you get a chance and you want to learn some basic developmental genetics, it's a great place to start. What's the Evo part of this EvoDevo paper? There isn't an Evo part. Yeah, in my little video about this, that was one of the first things I said is, yeah, this is a developmental genetics paper. It doesn't directly address evolutionary issues, but maybe that's a good place to start with this is what exactly about it contributes to EvoDevo. And what the paper does is, as you read it, you'll discover they just sat down and did saturation mutagenesis. So what does that mean? That means they calculated the number of flies they would have to treat with this particular mutagen to get a likely hit on every single one of the genes involved in the processes they cared about. And so they cared about early pattern formation. How do you set up the pattern in the fly, in the embryo, and in the early larva? And so they were just going to go in there and blitz it and just zap every single gene they could, and then look for the chance events that would produce genes that knocked out particular things in early development. And why is that important for EvoDevo? Well, because as they explain, what they're trying to do is get a fairly complete map of what genes are essential in setting up the pattern in the early embryo. Let me emphasize that word pattern a little bit, because they're not trying to hit every single gene that's active in the early embryo. As I mentioned, things like genes involved in metabolism, they don't care about those. Other people could, and you might say, well, we're going to dissect all the metabolic pathways by doing a saturation mutagenesis screen for effects on energy production and processing of amino acids and whatever. This is specifically talking about patterns. So why do you have those particular shapes? Why do you put bristles in this place and that place and this other place, but not in this place over here? So we wanted genes that affected that. So saturation mutagenesis, in this sense, it's you hear about fishing expeditions in research where you don't know what you're going to get. You just go out and you look in a particular place and see what you're going to get. And I think that's perfectly legitimate science. That's good science. That's just exercising your curiosity and making detailed observations. So it's perfectly fine. Popper might not have liked it, but screw Popper. Anyway, so saturation mutagenesis, though, is not like just going on a lightweight fishing expedition. That's where you go out with dynamite and you blow up every square meter of the bottom of the pond, and then you see what fish you get floating up to the surface. So that's what Nüsslein-Volhard and Wieschaus were doing, is basically tracking down all the different possible genes that are involved in this. Now, when I started this, I was going to explain to you why this is relevant to EvoDevo. It's because once they got that framework, they found all the pieces, and they've got the sequence for all of the pieces, and they know how to dissect those pieces further. So you've identified a gene, you can then sequence the gene. You can then make probes, for instance, just simple monoclonal antibody probes, whatever. Once you got all those tools in place, then it's time for the evolutionary part, the comparative biology part. So what they did is formulate all these tools and probes and useful measures of early pattern forming behavior. And then they shopped those out to lots of other people, and they adopted those and started looking for things. So, for example, way back when I was working in Utah, working with Scott Stachel, the late Scott Stachel, unfortunately, we did exactly that. We wrote off to Nüsslein-Volhard's lab, and we said we're really interested in these genes, gooseberry and bicoid and all these things that are involved in early establishment of the axis of the animal. And we asked, can you send us some probes? And it was amazing, they did. This is, lots of people have really good feelings towards Wieschaus and Nüsslein-Volhard because they were so generous in providing these tools to others. So, yeah, they just sent these all off to everybody who asked for them. We were able to look in zebrafish for things like goosecoid and other genes are involved in early pattern formation. So it was just a very productive thing to be able to do. And I wouldn't be surprised if one of the reasons that they won the Nobel Prize is simply because politically they had so many friends in the science community. Let's see. Oh, we got questions here. Let's see. How did they do this? Did they do full sequencing of each genome of larvae? Oh no, this was 1979, 1980. You couldn't do that. In those days, what did you do? Well, there were cytogenic markers. So you'd look at the larval chromosome, which you can see the banding patterns, and you can figure out perturbations in that. A lot of the early Drosophila work was done that way, where you've knocked out something, you go look at the banding pattern and you look for deletions, and you can map that out pretty well. So no, they start there, and then the goal is, of course, to develop a probe that you can use to stain for a particular protein, or particular gene sequence. And then you pull that out. And yeah, you move those along, and then you do it piece by piece. So you don't do full genomes. You didn't. You could now, I suppose, but you didn't then. You just use those probes to find the relevant portions of the fly genome to sequence. Amazingly, one of the things you can do with Drosophila is, you think you know where the region is. You can actually go in and physically chop the chromosomes, those polytene chromosomes that you find in the Drosophila salivary glands, and pull out physically a little region of the genome and put that in there. Let's see, do we have a fossil record for the ancestors of Drosophila? No. Drosophila is pretty tiny and fragile, so no. Oh yes, credit where credit is due. Trillion23X says Wieschaus and Nüsslein-Volhard got lots of research materials, fly strains and techniques, and genetic methods from Ed Lewis. Yes, Ed Lewis did some amazing work. He was a guy who was he was doing classical genetics, where everything's very abstract, and you do these crosses and you do the combinations, and you just try and sort out with pure genetic methods these different variants that affect the feature you're interested in. It's a tremendous amount of work, and Ed Lewis did a lot of that in the 60s and 70s. There's a good reason he's one of the co-winners of the Nobel for this, is because he contributed so much. So yeah, there's a lot of tools floating out there. This is a common theme that in science, we see further by standing on the shoulders of giants, and you really can't separate Nüsslein-Volhard and Wieschaus from Ed Lewis or any of the prior researchers. Nüsslein-Volhard, who she worked. She was a postdoc in, what's his name's lab? Maybe somebody remembers. The guy who did so much embryology of early insect larvae. So she had a really strong embryological background as well as a genetics and molecular biology background. And it's combining those two that really makes major progress in these sorts of fields. Trillion also mentions sharing relative generosity was an ethic in Drosophila research set by Tom Morgan and Sturdivant. Yeah. This is one of those things I really wish more labs would do, is just open sharing of everything they've got. That's how science really proceeds. And when I was working in a zebrafish lab, that was also the ethic there, is yes, you get a result, you share it with everybody. No secretiveness. All completely open. You share it at meetings, you share stocks, it's all the real thing. The nematode community is also that way. So is there any community that is not that way? If there were such a thing, it would wallow in obscurity and disappear. So no, if you want to be right at the forefront of all this cool stuff, you definitely want to do all this sharing of information. Okay, let's see, what else did they talk about in this paper? There were a couple of points I wanted to make that I didn't in the video. One is that the paper does a fairly spirited defense of model systems. That they make a point that if you want that raw depth of information, if you really want to get into it, you definitely want to do a model system where you got a group of labs that are all working on the same thing, just exploring different parts, different aspects of it, and plowing deeply into it, because that's the foundation you can build on. Once you've got all this information about Drosophila development, you can start looking in other organisms for the similarities and differences. But you got to have that starting place. So that was an essential part. The other thing I wanted to mention that I was pleased to see that Wieschaus and Nüsslein-Volhard also mentioned is this is a saturation mutagenesis experiment where you want to try and knock out every single one of the genes that affects the process you're interested in. And that was doable in Drosophila because Drosophila is a really, really purified organism. It has gone through so much selection for streamlining its development that there isn't a tremendous amount of superfluous variation or duplication there. You look in something like a zebrafish or any vertebrate, there is a tremendous amount of duplication in the genome. So you could do this same experiment in zebrafish, but then you would miss a whole lot of genes because gene A might have a gene A prime, a duplicate copy that's a slight variant of it that does pretty much the same thing, and knocking out one of those genes, the other one just steps forward and takes its place and you don't get an effect. So yeah, you want something that's stripped down like Drosophila. I was also sort of on the side of some of the work that Corey Goodman was doing back in the 80s and 90s, where they were going to try the same thing with neurodevelopment. So they were asking about the development of the nervous system in the fruit fly. And there was so much duplication. There are so many things where you knock it out and you get really subtle effects, maybe, if you're lucky. Stuff that requires detailed behavioral screens to puzzle out. But if you knock out two particular genes at the same time, you get dramatic effects on nervous system development. So that part of the fly is probably much like that of other organisms in that there's a lot of built-in duplication, and so you can't get as simple a knockout. You can't knock out a single gene and see a discrete effect. Oh yeah, it's mentioned in the questions. Biomed research is secretive, isn't it? Yeah. If you've got lots of money behind something, if you've got lots of money behind drug research and so forth, that really kind of spoils the science. It's kind of unfortunate. Pharmacology researchers, when they're at the top of the game and they're involved in industry, can make buckets of money. And that means you've got incentive to be hush-hush about it, because somebody else might scoop you on it. Yeah, when I lived in Philadelphia, there were a huge number of pharmaceutical companies and biotech companies, especially in New Jersey, and some of them, just the conspicuous waste was embarrassing. There was one pharmacologist I knew who had his car gold-plated because he could. That's just such a waste. Let's see, some parts of astronomy can be secretive. Really? There's not a lot of money in astronomy. That would surprise me. Let's see. Would you say that a caterpillar is a butterfly fetus outside the womb? Is that a useful way to think of it? I don't know. Yeah, I wouldn't think of it that way, because these holometabolous insects like that are going through discrete phases in their development, where they can follow very, very different lifestyles. And they're basically whole different organisms that are exploiting a different energy source, for instance. One of my colleagues here is an expert in mites and ticks and things like that, and she's studying, life cycle of mites, and they're just unbelievable. They might go through like 7 discrete instars, where in one they're a vegetarian, they're living on algae in the water, and then a little later they metamorphose into a carnivorous larva of some sort, and then they metamorphose again, and maybe they're once again back to the vegetarian lifestyle. It's all tied to seasons of the year and so forth. But it gets pretty elaborate. And yeah, it's like one organism with multiple potential life forms built into it, and it's kind of groovy. Oh, the part of astronomy, okay, getting back to that question of why astronomy would be so valuable is the part where the satellites are looking down, not up. Oh, now I get it. Okay. Yeah. Yes, and young scientists make a choice between doing open science and making the big bucks, I guess. This is also it's also a choice point. Yeah, some fields you could easily transition from doing pure research, just tinkering for the curiosity of it, into developing skills that are useful in pharmacology. So like, if you're good at tissue culture, yeah, there's nothing there. They need those people over there in the big pharmaceutical companies. So there's lots of ways you can go. It's actually a good point to bring up, because it's one of the things we have to do as educators at this university. Is you get these young students who come along and they're as limited in their mindset as I am, and they think, oh well, I'm going to spend my life doing research and get a professorship and just do this all the rest of my life. But no, there's a lot of other stuff you can do with a biology degree outside of teaching more biology. Let's see. So Blake points out, if you spot a supernova, you want everyone to see it before it fades. You tell everyone. If you find a new Kuiper belt object, it's not going anywhere, so you can sit on it and learn as much. Yeah, okay. This is true. Are there analogs in biology? I'm trying to think. Is there such a thing as... No, there's not really. A friend of mine was doing research on model systems, and she switched from insects to doing annelid larvae, marine larvae, and marine worms. And there was no incentive to keep it secret until you could suddenly explode onto the scene with all this dramatic stuff. And no, you want to keep putting out there as much as possible. Yeah. I'm also thinking of for instance, the King Lab, where they do work on quantiflagellates. Yeah, that was just exciting stuff from the very beginning, because I just kept churning it out there. Oh, engineers. Yeah, there's another interesting choice is, are you going to work in military electronics? There's nice stable income there. Get employed by a defense contractor, and oh yeah, you're probably doing pretty good. Way back when I was in graduate school, one of the professors at the University of Oregon was tapping into that defense money. It was, in some ways, it was kind of hilarious. He was studying grasshopper brains, and so he had a proposal to extract the brains from insects and put them in a little life support system and hook the terminal nerves up to various sensors in missiles and things, and you could train the grasshopper to fly the missile into a target. Yeah, I guess. Okay, that's. I think it was a bit of a reach. I wouldn't trust a grasshopper brain to sort out good targets from bad targets, but also that whole idea of taking in money for that. A lot of insect researchers too are getting pesticide money, especially in neuroscience and invertebrate neuroscience, because most of those pesticides that we throw on insects, they target specific details of the insect nervous system that do not share homology with those in vertebrates. So you go looking for something that's weird and unique about an insect nervous system, and then you target that with some kind of drug that does next to nothing to humans. Yes, bug-guided missiles. What's wrong with that? Again, this was back in the 80s, okay? And back then, the idea was, rather than building nice little tiny integrated circuits that did all the fancy stuff of visual processing and guidance, you got an insect. It's already got a brain that is expert at visual guidance and signal processing and steering the insect body. So what if you could just take that out and stick it in a machine and have it do it for you? Yeah. Okay, let's see what is. Yeah, out of all the things, yeah, and you could look at it as this is a money sink for the military. So they're actually throwing money into invertebrate neurobiology research. And if you were in the know, you would be sitting there chuckling because they're never going to get anything out of this. But then you never know, you might get that breakthrough, and all of a sudden, you're doing all kinds of cool things for the military. Ugh. I shouldn't shudder. My son is in the army, but he's doing a lot of good things. Insect slaves in a human nation. Yes, that could. All right. Ask me questions. Otherwise, I'm gonna talk some more about this paper. There's all kinds of good stuff in here. This I think is one of the really important secrets of what they did is they homed in on a system that was comprehensible. Look at an insect, look at a spider, for instance, and if you start with an adult spider and you look at it very closely, oh man, they are complicated. There are so many little details, and in the mandibles and the chelicerae and the eyes, all this kind of stuff. These bristles, they all have specific patterns of bristles in various places, and it becomes kind of overwhelming to try and sort those out. And what Nüsslein-Volhard and Wieschaus did that was brilliant is focus on what is analyzable. So, this picture right up here, this is the larva. So you've got methods for clearing it. I think they use the same stuff I use, halocarbon oil, which doesn't suffocate the animal at all, because halocarbon oil, lots of oxygen saturates that pretty easily. So you're not suffocating the animal. You're not damaging it in any way. You can have a live animal even. It also works with dead animals. But you just cut it with this, and it has the additional thing that has roughly the same refractive index as the cuticle of the larva. And so you can look right on through. So that's what you're seeing here, is that dark thing is, you get some nice dark field optics, and you just kind of look at it and you can see this really distinctive pattern. Like there's the jaws up there at the top. Each segment has this row of bristles. If you look more closely at them, the bristles actually have segment-specific patterns. So you look there, and that looks different from this segment over here. So you can identify segment by segment. You can see various organelles in there. It's a really nice system because this is comprehensible. That's the nice thing about it. A spider face is not comprehensible to a human. And we've reduced it to a small number of elements that you can look for variations in. This was always a problem with zebrafish for me, is that when you study zebrafish for a while, you get pretty comfortable with the morphology and you can actually recognize individual zebrafish. So it's like human faces. Look at, everyone is really good at spotting subtle differences, little differences in shape of the face, shape of the eyes, the eyebrows, the nose, the mouth. All these things add up, and there are things you can look for. But unfortunately, human face is so complicated in that sense, you get lost in it. Where are the genes that affect all the details of the face? They're all over the place. And same with the zebrafish. Yeah, there's a lot of subtle variation there, but it wasn't very approachable. For one thing, it's really subtle, but for the other thing is these subtle things are often polygenic. There's lots of stuff that controls them. So you can't just knock out a single gene and say, oh, well, that's responsible for the shape of your nose, because it's not. There's a whole bunch of genes responsible for that. But when you look at something like that pattern, that is so clean, and you're just looking for genes that perturb that particular pattern, yeah, that was a good move on their part. Once again, credit goes to Nüsslein-Volhard and Wieschaus, but it also goes to their advisors, their history of education, all the people who did this prior work that led up to it. Yeah, you never know what's going to be really important further down the road. Okay, I see Rory Cat's made it here. What are you doing here? You don't know anything about development. You're a cat. Are zebrafish good to eat? Oh, that's something Rory Cats could have some expertise in. Sure. I've eaten zebrafish as anchovies on pizza. So yeah, they're fine. They're small, but I bet you a cat would get into zebrafish for dinner. They'd be pretty tasty. Yeah, and I suspect they're kind of oily, just like, herring and small fish like that, sardines. So there's probably a lot of good nutrition in a zebrafish. You just have to eat a lot of them. So, yes. Think of zebrafish as fancy anchovies. Okay. Oh yeah, also in this paper they give you an introduction to all these different maternal and zygotic mutants. Where'd I put it? Oh, there it is. I mentioned this book. This is a great place to start. No, really, if you don't want to dig right into a technical paper by Nüsslein-Volhard and Wieschaus, this is a really excellent summary of their results. And it goes through all the phenomena. Somebody mentioned butterflies being good. Yeah, butterflies are good for studying pattern as well. Sean Carroll has been doing some work on butterfly pattern formation. Yeah, this is Ed Lewis's bithorax mutant. So, yeah, another interesting thing in the pattern. Oh, and this Nüsslein-Volhard and Vischhaus paper doesn't really get into this so much, but imaginal discs. This is another cool aspect of working with flies, is there in the larva there are these little knots of tissue that at metamorphosis are going to expand and evert and form the cuticle of the adult fly, and all the other associated structures that are there. So this is the wing disc right there. So it's a flat sheet. You can see kind of a spiral pattern in the middle of it. If you look really closely, if my camera will focus that closely, and it actually just sort of poof, pops out to form the wings of the fly. When I was an undergraduate, that's actually what I was interested in, is looking at imaginal discs. And among the things I was doing way back then is trying to figure out how the connectivity of the nervous system of the wing establishes itself. And one of the secrets is, it starts out really small with everything close together. And then you don't have to grow very far to reach the central nervous system. Okay, the name of the book is The Making of a Fly by Peter Lawrence. The Genetics of Animal Design. Yeah. So, I mean, this book came out not long after they did their work and before they won the Nobel Prize. And it's just an example of how quickly progress was fueled by these initial discoveries by Nüsslein-Volhard and Vischhaus. Oh, there's another. There is so much beauty in a fly. Okay, what is this? This is the retina. So we're looking at the formation of the ommatidia of the compound eye, and this is gorgeous stuff. Yeah, I used to work with a guy who was kind of nuts, but he did a lot of early work on. His name was Tom Hansen. Did a lot of early work on development of the ommatidium, and he came up with a catchy name for it. He called it a neurocrystalline lattice, and it really is. It's just these beautiful patterns repeating over and over and over again, and you can find mutants that affect specific segments of this. So let's see, what can you see here? If you look in the center of the ommatidium, there are the 7 photoreceptors, and they have a specific orientation, and they have a specific arrangement, and you can mutate those in ways to disrupt that. They also have specific functions, specific wavelengths that they respond to. It's a really repetitive pattern. Also, what's kind of cool is that the equator of the eye, so the space across here, is a point where everything flips. So you can see that there's kind of a trapezoidal arrangement to this. And if you go below the equator, it flips upside down, and all the ommatidia are upside down relative to this. So it's a very detailed, very intricate sort of process that builds that. Do they talk much about the morphogenetic furrow? They must. Yeah, and that's another thing where it's been worked out in detail exactly how it all occurs. That's the eye antenna disc up there at the top. And then below, what you see is there's actually a physical wave that sweeps across the sheet of the prospective retina, where all the cells are going through mitoses and they dip down, so they build a little furrow, and then the cells next to them dip down. And, it really is like the wave performed at sports ball games, okay? And you get this wave just sweeping across, and as the wave passes, the individual cells spontaneously crystallize, even organize themselves into these structures to make up the retina. So the Nüsslein-Volhard paper is all about early pattern formation. It's exclusively about early pattern formation, the stuff that's being set up in the embryo. There's also a whole bunch of stuff in this book, The Making of a Fly, that also talks about later events. So it's, let's see, I always wondered how the compound eye works. How many photosensitives? There's 7 photoreceptors in each ommatidium. Yeah, and they have a specific arrangement. Yeah, it is very... Okay. Oh, actually, this is a good point. Phoenix Marine says that looks very complex. Is it? Is it very complex? I don't know. How do we define complexity? Because on the one hand, it's a very repetitive pattern, and it produces an arrangement of photoreceptors that are predictable and precise and occur in every single animal. Yet if you look at the vertebrate eye, is it more or less complex? Because we don't do that. We have kind of a salt and pepper arrangement of these different cell types, that, for instance, the different color receptors. We've got 3 different color receptors, and we got just an intensity receptor, 3 cones and 1 rod. And those also have significance in visual processing, but they don't have this really repetitive, precise lattice. They're just kind of scattered, and in a way that produces, a nice salt and pepper pattern of everything, so it all works. And then what has to happen is that the brain has to reconstruct that spatial information from the signal information coming back into like the lateral geniculate and so forth. Which is more complicated? I don't know. Yeah. I kind of suspect that the compound eye, of Drosophila at least, is simpler. I can't give you any kind of hard measure of that. But the reason I think that way is because flies evolved to rapidly develop, and so they've kind of streamlined everything. And in this case, this is a streamlined eye. It develops quickly, and it builds a consistent structure. So, I don't know. You tell me. Okay, ADX is complexity, smumplexity. You can only measure that stuff arbitrarily. How many polygons do we need to make it look good in the videos game? Yeah. Yeah. How many polygons do you need to make it look really good in a video game? Because then you look at something like Minecraft. They have greatly minimized the number of polygons they need to process, right? It's fairly simple and straightforward, but it makes for a streamlined game where you can go in and just do the things you want to do. And is that more complex than arbitrary number of polygons? Huh? It depends on what you're trying to do. Oh, I'm sorry, Donald. Your vision is getting so bad you'd probably settle for a fly's eye right now. Well, again, there's differences. That your eye is good for resolution and distinguishing things, better than a fly's eye right now. I'm pretty sure. I know my vision has been going downhill as I get older too, but I still think I can see things better than a fly eye. However, a fly's eye is really good at vision of moving objects. It's really quick to pick up on changes. Yes, Gering's lab. I should talk about one of Gering's papers on one of these videos. Yeah, so that there's Gering and PAC6 and Eyeless. That not everything came out of Nüsslein-Volhard and Wieschaus. They did the foundation. But all the cool stuff that comes along later, a lot of it is coming out of like the Gering lab. So yeah, the whole idea of inducers of complex structures. So how do you get, I just mentioned how you form the different components of the retina with a morphogenic furrow moving across and all these events occurring. But an even earlier question is how do you set up an eye field? What genes are important for that? And Gehring did things on, ILIS, PAC6, all these affiliated genes that are essential for establishing that early eye field. It's also the same genes that work in establishing our eye fields. No, Blake, that T-Rex vision stuff from the Jurassic Park movie was just made up. It wasn't real. Yeah. The whole. Oh, it was because of frog DNA, wasn't it? Oh. No, that's. There is so much bad science in Michael Crichton books and movies. I can't read them. I can't watch them. They just drive me nuts because that's not how anything was going to work. There's a good question. Are spider eyes more fly-like or human-like? And are all eight eyes designed the same? No, they're not all the same. And they're both. That in our last common ancestor of vertebrates, flies, and spiders and all that, there were actually two kinds of eyes. Just two. That there's just a pair of eye fields on the face, and they develop in different ways. So one set of eyes is more sensitive to light levels and is important in things like circadian rhythms, and the other is modified by evolution to form image-forming elements. And the interesting thing is that in Drosophila, it's these lateral fields that form the compound eye and are the image-forming elements of the fly's sensory vision. And then there's also a central, what were they called again? Suddenly I'm blanking on the name. There's also a couple of other small eyes in fruit flies and other insects that are important in just general detection of light levels and the levels, direction of illumination and basic stuff like that. Spiders start off the same way with two kinds of eyes, and there's one median set of eyes that are going to form the pattern forming eyes, the big round things that you see in jumping spiders. Those are more like a camera eye, like ours, where they've got an outer lens and a retina that contains a vision-forming element at the back of them. And so they're more like us in that sense. Yes, and different spiders have wildly different eye setups. And there's some interesting stuff going on in an evolutionary sense, because they got this battery of eyes, okay, and each of those eye fields does subtly different things. So what you see is different species of spiders amplify one or the other into vision forming elements. So I was reading a paper about this just the last week, and I'm trying to remember the details. But like some of the the ocelli, that's the magic word I was trying to remember. And some of them use these central pair of eyes, which have one kind of photoreceptor in them that is particularly good for sensing light during the day, and the others out here have a different photoreceptor, and you've got some nocturnal spiders that instead of the same eyes that jumping spiders use, they increase the size of these lateral eyes and migrate them around in different ways, and use those for primary vision. So like ogre spiders and jumping spiders, they both have great big eyes in front, but they are not homologous eyes. They arise from different parts of the set of eyes. Some spiders have 12. I don't know any spiders that have 12 eyes. Yeah, eight is the canonical numbers. There are some that have 6, and there's a few rare species that don't have eyes at all. Was there some sort of wild event where evolution separated a bunch of spiders from the rest of the crowd? No, what it is, well, it's spiders are diverse, and different groups of spiders may be separated by 300 or 400 million years from other groups of spiders. So there's a lot of stuff going on. One of the things that's really easy for a different spider lineage to do is to, for instance, fuse eyes. So you get a pair of them fusing, so then you get a 6-eyed spider. Or just losing them all together, or emphasizing one eye, one kind of eye over the others, like you see in jumping spiders. So, was there there have been multiple speciation events, and different lineages of spiders have evolved in different directions. I mean, some of the more significant differences, though, are feeding style. So jumping spiders are, they're hunters. And also wolf spiders, things like that. They just wander around using speed and ability to jump and good things like that to catch prey. And lots of other spiders are more stationary. They're ambush predators. So there's lots of differences there. Ogre spiders, do their eyes have layers? Oh, well, in general, do they have layered retina with multiple cell types? Yes. There's also some with some really sophisticated optics. So jumping spiders, for instance, if you ever dissect one, it's kind of cool. They do not have spherical eyes, like we do. They've got cylindrical eyes. Their eyes actually stand way back into their head, and they're more analogous to telescopes, little telescopes on little swivels that they can move just a little bit, but mainly they turn their head to aim them in particular directions. Yes, there are blind cave-dwelling spiders. Look at this, we've wandered off into the topic of spiders. What is wrong with you people? Oh, it's all good. It's all good. So yeah, I should do something on just spider eyes. There's some amazing work that's been done on spider eyes. Mike Land is the guy I first started working with spiders on. Mike Land was a visiting professor at the University of Oregon, and he taught a sensory physiology course, which was wonderful because this was a really wild kind of laboratory course in that he just sort of said, hey, here's some cool stuff, go explore it. And so he introduced us to jumping spiders, and for a time there, all the graduate students spent all their free time wandering around collecting jumping spiders off of walls outside and taking them in and doing experiments on them. And yeah, we dissected some. That's mean, but we also did things like super glue stuff to them. So for instance, one fun thing we did was we'd super glue them to a thread, and just so that we could control them, and we'd make these rings out of tissue paper. And this was actually interesting for studying what the spider was interested in. Is you have this ring of tissue paper, and you get a spider on a thread clinging to it. They don't want to let go of it. And you put visual stimuli out there, and they will track it And you can see it really easily because as they're scampering along on this ring of toilet paper, they would turn. You could actually see them turn the ring in the direction of whatever had caught their attention. Huh. You've got references to spiders up to 12 eyes. I will check my, over there, not on the shelf I talked about yesterday, but on another shelf, I've just got a big collection of books about spiders. And I will just have to look through there. I don't recall ever seeing a 12-eyed spider, though. That would be neat. Yeah. I'm hoping with my new camera, my new camera lens, I'll be able to get some really good close-ups of spider eyes. So I will try that later today. See what I can see. Spider Shrek. Okay, I don't get the reference. Yeah, you can't trust the internet. The internet, it's the internet of lies. There's all kinds of garbage floating around. Uh, but, I'll double check. Spiders are extremely diverse. They go in all kinds of different directions. I could imagine a developmental mechanism where you get more than 8 eyes. I just don't know if any spider has ever implemented it, because they all start off with 2 eye fields, and one of the eye fields splits into 3, and then the other one. Anyway, there's ways it could happen. I just don't know if it's happening. That would be interesting to discover. Yes, Google has holes and takes shortcuts. Even Wikipedia is not entirely trustworthy. What can you do? It's one of the reasons everyone ought to go to school and learn critical thinking and then learn how to do proper research. One of the things I keep emphasizing to people, to my students too, is you can't figure things out from the internet. You might get a superficial understanding of it, but the difference between the people who have deep knowledge of something and the people who have YouTube knowledge of something is whether they read books. So read more books. Okay, another question. Let's see. Oh, yeah, Nephilim free. I don't care. He's an idiot. Yeah, he's just not worth talking to or about. All right, so 8DX is wondering how, now we have automatic genome sequence, is it easier to do those mass mutation studies than before? That's an interesting question. Let me think. Well, no, because now we're getting into real fishing expeditions. You could say, oh, I'm just going to throw a mutagen on something and then I'll sequence the whole genome and see what I find. And most of the information you'll get out of that kind of sequence is redundant. It's just the same thing everybody else has figured out before. So why would you waste your time doing that? You want to have a more targeted approach. There, for instance, the zebrafish genetic screens were much more tightly focused. Let's see, George was doing all this work on zebrafish visual systems, and so he just focused on finding retinal mutations, and that's good enough. Okay, so it narrows the field down a lot. Chuck Kimmel was doing all this interesting work. I don't think it ever panned out. I just remember, every week there was the ongoing screen for mouth nacelle mutations. So there'd be a new batch of embryos that had been treated with mutagen, and they'd line them all up, and they'd look through and they'd look for variations in the decussation of the mouth, this big hindbrain neuron that grows into the spinal cord. It's so big that you didn't need to stain the animal. You could actually visualize it with just the microscope. And yeah, we were in there every week looking at those things, and I don't think he ever found a mutation that. Okay. Oh, would that cancer biology book you mentioned yesterday be a good start for a physicist to actually learn the subject? Oh, no, probably not. It depends on how knowledgeable you are about cell biology. Uh, it's probably not a great starting point. Um, you know, I have another textbook I used. So as I mentioned, I used the Weinberg book in my cancer biology course, and we quickly decided it was too much. It was just overwhelming the students. So another year I tried a different textbook. I'm trying to remember the name of it. But there was a more, maybe I will drop you a note, Blake, and let you know what a better book would be for starting on this subject. Yeah, the Weinberg book it's got everything. It categorizes all these different things that can happen to produce a cancer cell and breaks it down, and then goes into excruciating detail on everything about it. And yeah. Okay, 8DX has a question. I mean, looking at what the result was for each of the mutations. Yeah, so what Chuck was specifically looking for is mutations. So it's a decussating neuron. That is, you get one on each side of the hindbrain, and they send an axon across that crosses in a nice little X and then proceeds down the spinal cord. So specifically, they were looking for the any variations in that. Could you find mutations that screwed up the midline in some way so they didn't cross? That, this actually this was of interest to me. It's actually what I did with grasshoppers when I went off to a postdoc, is we had neurons that decussated that crosses at the midline, and we tried to find the cues that led them across the midline. But yeah, but we didn't find anything. Oh, the Peter Lawrence book costs $50 on Amazon. Oh, I'm sorry to hear that. Yeah, and it's from 1992. It's a classic. They should come out with a Kindle edition. That would be nice, but then they probably charge just as much for it. Yeah, that was a problem with a lot of the books I mentioned yesterday, is they aren't cheap. They're generally textbooks, and textbooks, it's pretty common for them to cost between $100 and $200 each, which is just not right. I kind of hate that. But on the other hand, the place to look for those is to look in used books, because what happens is those books get used up for a semester and then get passed on. Through because the publishers churn so much and come out with new editions, they get cast off, and then you can buy the used books much, much more cheaply. I think our cell biology book I saw was something like $20 if you bought an edition from like 2 editions ago, used. So that's one way to go. Unfortunately, this doesn't have that kind of thing. I don't think there's been another edition of this, because it's so comprehensive and thorough. And yeah, it's just going to be hard to find this one used. But maybe you could. Yeah, you can't have mine. This is mine. It's a really useful book. I can't tell you how useful this was. It was just so much stuff in there. And as I mentioned, I did use it as a textbook a couple times when I taught invertebrate developmental biology, because that does everything, covers the whole field. And you use that textbook, and you have some supplemental papers, and you got a pretty detailed course. Okay, let's see. Oh, more questions. So there's a lot about getting genetically engineered goats to produce spiderweb protein. It's been done. I know they're doing it, and they've got it. Um, I don't know where it's gone since, though. I would think that a big part of the problem is that it's not simply finding this glop of protein. Spiders spin it very precisely. It's being extruded out their spinnerets in very specific ways, and so you'd need to have some way to also produce that cable structure that they do. So that might be a major obstacle. Let's see. Rory Katz did Google searches. Oh no, Google sucks. But no, they all suck. But anyway, yeah, there's websites ripping each other's text off, and it spread. The 12-eyed spider story spread as a result. Again, there's so much diversity in spiders. I will have to consult my detailed taxonomical texts and find out for sure. No, the goat thing was not an utter failure. Come on. Tired pun, too. But anyway, yeah, no, it's good stuff. Being able to mass produce specific proteins is one of the virtues of the modern age. Look at for instance, insulin. That's a simple one, but yeah, producing spider silk proteins, and and then finding a way to process them appropriately so that you could build spider silk cables. That would be kind of awesome. There's this whole field of material science. It's amazing. It's got great stuff that that's where I think a lot of the surprises we get in technology come out of, is material science. Okay. We need spider goats. Yes. Yeah. Personally, what I would like to see is being able to engineer more legs than 4 in a vertebrate. It's just so fundamental to the tetrapod body plan, though. It's just not happening, unfortunately. Let's see, we're spending a lot of time talking about spiders. Oh, this is. Let me mention one other cool thing. The paper talks about techniques, which are kind of awesome. I love this technique. So this is their procedure for producing large quantities of identifiable embryos. So you got these test tubes. That's what you raise your adult flies in, and then you just put that down on a petri dish with this special agar they make with apple juice and things like that. So it's a good medium for the flies, and the flies just crawl around on it, and they lay their eggs right there in the spot. So you get these nice patterns. You can actually just go in and analyze each one of these for different mutations. Oh, Donald, you're asking hard questions. The Evo process proposed for Metamorphosis. Are there intermediate forms, as for, say, evolution? Ooh. There's... Metamorphosis is one of those areas where there's a whole lot of wild, creative ideas floating around. I don't know. Some of it's kind of off the wall, but yeah, I don't know that there is. You look at a fly and it's got those imaginal discs that are going to form the adult cuticular structures. I don't know what the intermediate step for that was. Maybe there wasn't one. Maybe this is a case. Yeah, there must have been some intermediates. I'm not sure exactly what they would have been, though. So in some ways, maybe the precursors are just the process of molting. So you look at how flies and spiders and everything molts. There's a lot of work that's been done in this. You got the outer cuticle, and they actually form an interior cuticle right underneath it. And that develops, thickens, and then they split the outer cuticle and they emerge, and then the new cuticle tans and forms rigid structures and so forth. So you could imagine something like that happening with a larval form having an instar that formed the cuticular structures for the next stage, and that cuticle was subject to all kinds of different selection pressures, and leading to some interesting conclusions there. I don't know that much about metamorphosis, though, so I can't say. Yeah. I will say that there are people who talk about, the hopeful monsters idea. This is saltational. I don't really believe that. Especially when you look at the different lineages of insects, and you've got the hemimetabolous and the holometabolous insects. The holometabolous must have evolved out of the hemimetabolous insects. So there was a stage where there was a lineage where you're getting progressive elaboration on the adult form. So baby grasshoppers look a lot like adult grasshoppers, but there are also significant differences. So start from there. Okay, we've been talking for over an hour. This would probably be a good place to end this all. How many people do we get showing up today? Let me see. We got up to 24. That's pretty good for Boxing Day. Yeah. You guys must have better things to do, right? Oh, that's a good... Somebody fed an insect ancestor chicken after midnight. Hey, presto, metamorphosis. Yes, you watch Gremlins. Okay. Yes, the Gremlins theory of the evolution of metamorphosis. And metamorphosis had to have evolved multiple times. I'm sure it has. Yeah. So different organisms must have evolved different patterns, radically different patterns. Another factor that might come into play here is so many of these metamorphic events happen with transitions to different environments. So, aquatic larvae evolving into flying adult insect, yeah, that kind of thing. Have I heard of the duck hand phenomenon? No, I'm afraid to ask. What is the duck hand phenomenon? Tell me quick, because I got to end this soon. I got spiders to go take care of and photograph. So what is the duck hand phenomenon? It's going to keep us hanging, isn't he? Or he's typing frantically with his little kitty paws. Must be slow. Oh, we get a link. Okay, let's see. Show that link. Oh, error. Oh, it's not letting me show your link. I know how to get around that, though. Let's see if I can. It's a duck hand phenomenon. I will describe it to everyone once I get it.
In over 100 years, we've never missed a payment. Guaranteed monthly income for life.
Okay.
With HP, it's easy for IT to get
I'm
the right
getting the usual
devices to the
YouTube
right people
commercials.
and help employees.
One of our subscribers recently asked a really interesting question about duck
Human
hybrids, and I wanted to try to answer it today and talk to you about some of the weird
Duck
things that
human
we've seen here on
hybrids.
our farm. But first, release the Quackers! Right here, you actually see our interesting little flock that we have.
Well, I
that
will
like
let
to
you all
call the
go.
Parks
I'm
and
going
Rec
to watch
gang.
about
The little duck,
hens.
the little brown duck that you see there with these
Then I'm going
geese.
to go play with
Her name is
spiders.
Ron Swanson, like the Parks and Rec
And
character.
Maybe I'll post something about
We
this
hatched
later.
these birds out all together at the same time
Okay, so it's
earlier
been
this
fun,
spring.
everyone. Let's end it here, and we will talk to you all later.